Let's now study the digestion of maltose. What are the glucose test strips? As their name indicates, these are special strips used to indicate the presence of glucose. How do we use these strips? First of all, we dip the strip into the test tube that contains the food to be tested. Then, we compare the color obtained on the strip to the range of colors on the container. The darker the color obtained, the more glucose the food contains. Let's now move to the experiment. The substrate is maltose, the juice is the intestinal juice. Interpret the result of the experiment. After dipping the glucose test strip into a test tube containing maltose and intestinal juice, the color of the strip turned into purple. This indicates the abundance of glucose. There are 5 enzymes in the intestinal juice. Lipase, that transforms lipids into fatty acids and glycerol. Erepsin, that transforms proteins into amino acids. Sucrase, that transforms sucrose into glucose and fructose. Lactase, that transforms lactose into glucose and galactose. And maltase, that transforms maltose into glucose. So under the action of maltase, each maltose molecule will produce 2 glucose molecules. What are nutrients? Nutrients are small and soluble molecules present in the small intestine and resulting from the complete digestion of food. Nutrients include glucose, amino acids, fatty acids, glycerol, water, vitamins, and mineral salts. Note that water, vitamins, and mineral salts don't undergo digestion because they are already simple molecules. Now let's sum it up. Digestion is a set of chemical and mechanical phenomena that takes place in the digestive tract and transforms the complex food molecules into nutrients. In the mouth, where the pH is equal to 7 and this is a neutral medium, the mechanical digestion includes mastication or chewing. On the other hand, the chemical digestion includes the transformation of starch into maltose under the action of salivary amylase. In the esophagus, the mechanical digestion includes the peristaltic movements, and this is done by the contraction and the relaxation of the muscles of the esophagus. This will help food keep moving along until it reaches the stomach. There is no chemical digestion in the esophagus. In the stomach, the mechanical digestion includes churning. On the other hand, the chemical digestion includes the transformation of proteins into peptides under the action of pepsin, which is secreted by the gastric gland. In the small intestine, the mechanical digestion includes peristaltic movements, as in the esophagus, in addition to churning. The chemical digestion is carried on by intestinal and pancreatic juice. The liver secretes bile into the intestine, which renders the medium basic and emulsifies the lipids. The pancreatic juice contains 3 enzymes: amylase to transform starch into maltose, trypsin to transform proteins into amino acids, and lipase to transform lipids into fatty acids and glycerol. As for the intestinal juice, it contains lipase that transforms lipids into fatty acids and glycerol, maltase that transforms maltose into glucose. So, by the end of digestion, we will obtain the following nutrients: glucose from the digestion of carbohydrates, fatty acids and glycerol from the digestion of lipids, amino acids from the digestion of proteins, as well as water, vitamins, and minerals, which are already nutrients. Describe the adjacent functional diagram in a short text. Note that while describing a functional diagram, you have to stick to the keys given in the legend. The stomach produces the gastric juice, which contains pepsin that transforms proteins into peptides. The small intestine produces the intestinal juice, which contains erepsin that transforms proteins and peptides into amino acids. And finally, you will be assigned to study table 5, page 4 on the subword guide and solve questions number 8, page 10 plus number 20. This is all. Hope you enjoyed the lesson. Study well and keep safe.